Good morning, everyone. Today we are going to dive in again to the word full of discovery. Let's dive into a word too small for our eyes to see, yet powerful enough to keep every living thing alive. A word where cells aren't just simple structures. They are highly specialized, carefully designed, and perfectly adapted for their roles. And today, our topic is about cell modifications. She knows all about science stuff, Professor Reese explains. Have you ever wondered what if sperm didn't have a flagellum? No tail to move, no way to swim, just stillness. If every sperm cell lacked that tiny whip-like structure, would it ever reach the egg? Would fertilization even happen? And if movement stopped completely, do you think procreation or reproduction itself would be still possible? Or would life journey end before it even begins? That is why we do have what we call cell modifications. Cell modifications are defined as the adaptations or changes acquired by the cell after cell division that aids the cell in various beneficial ways. And for today's topic, our learning objectives are define cell modification, enumerate and describe the three types of cell modification, characterize apical, basal, and lateral cell modification, and give example of apical, basal, and lateral cell modifications. First up, we do have apical modifications. From the word itself, they are found on the apical surface of the cell. Examples of this are, first, cilia. They are short, hair-like projections covering many spots on the cell surface. Their movement is synchronized, wave-like beating like R's rowing in unison. Second is flagella. It is a long, whip-like tail, usually only one or a few per cell, made of protein filaments. It rotates or undulates like a tail, propelling the whole cell forward. Third, we do have villi or microvilli. They are small, slender, vascular, finger-like projections that increase the surface area to increase absorption. And for the fourth one, we do have the pseudopods. They are the false pit that act as temporary, finger-like extensions of the cell cytoplasm. Their function is for movement and ingestion or the phagocytosis. And the last one is extracellular matrix or the ECM. It is compound secreted by the cell on its apical surface. Cell wall is the extracellular structure in plant cells that distinguishes them from animal cells. Glycoproteins is the main ingredient in ECM in animal cells. Second step is the basal modifications. They are found on the basal surface of the cell. The example of this are desmosome or hemidesmosomes. They are the anchoring junctions on the basal surface of the cell. They are rivet-like links between cytoskeleton and extracellular matrix components such as the basal lamina that underlie epithelia. It is primarily composed of keratin, integrins, and cadherins. Lastly, we have lateral modifications. They are found on the lateral or side surface of the cell, connecting them to their neighbors. First, we have tight junction or the ceiling fence. Structure is continuous, belt-like junction around the lateral side of the cell. It creates a water-like seal between two adjacent animal cells. Function is to keep liquid from escaping between cells, allowing a layer of cells to act as an impermeable barrier. Cells are held tightly against each other by many individual groups of tight junction proteins called claudins. Second, we do have adhering junctions or the wall bolts. The structure is belt-like or spot-like junctions with actin filaments anchoring neighboring cells. It holds cells tightly together. It provides mechanical support to maintain tissue shape. And lastly, we do have the gap junctions or the communication lines. Structure is protein channels or the connections connecting the cytoplasm of two adjacent cells. They allow rapid exchange of ions, nutrients, and signaling molecules. They coordinate activity between cells, and there is a presence of connection that allow direct exchange of chemical between cytoplasm of the cells. Whew! That's a whole city of modifications inside a tiny cell. But here is a summarized version of what we've learned today. But don't worry, we're not done yet. Next, we're going to turn all the cell moves into a catchy song, so your brain can dance along while it learns. There are 3 types of cell modification. Apical is the top, yes, it's the top. Lateral is the side, yes, it's the side. Basal is the bottom, bottom of the side. Second verse, here we go. Apical helps the cell to touch the word. Lateral holds the neighbors tight. Basal anchors firm down below. Keeps the cell steady, safe and right. Bye-bye for now, guys. See you again to a world full of discovery.